Now that we know this, we want to calculate the pH of different solutions. And that's going to be important because if we're going to do biochemical reactions, we have to control the pH and we have to figure out how we're going to do that. And in order to get to buffers, I have to just review with you how we do this, okay? So, for two cases, for strong acids and bases. For strong acids and bases, life is easy, right? It's easy because, for example, we only need two equations. One of the equations that we need is the ion product of water. Which, of course, is simply that the concentration of protons times the concentration of hydroxide anions must equal 1 times 10 to the minus 14 molar squared. The second equation that we will need is a mass balance of protons. Sometimes called the proton condition. Remember that as long as I'm doing a reaction in H2O, this equation, the ion product of water, must be satisfied. So, for example, to give you and again, the reason we can get away with only two equations, as you'll see in a moment, is we're going to assume that there's complete dissociation. So let's take an example. We're going to dissolve 1 millimole of hydrochloric acid into 1 liter final volume of H2O. Okay, now this is super simple because you know that hydrochloric acid will dissociate essentially completely into a proton plus chloride anions, right? And so if our initial concentration of hydrochloric acid is 10 to the minus 3 molar, most of it is going to dissociate completely. So there are two ways that we can do this, okay? First, we take equation 1. So what we're going to do here is this is our initial state. And we want to solve for the concentration of protons in the final state. So let's just use our two equations. We know, first off, that the concentration of OH minus is simply equal to 1 times 10 to the minus 14 molar squared divided by the concentration of protons. That's our first equation. Okay? And then we know from our second equation that there are two sources of protons. We know that H2O must dissociate into a proton plus a OH minus. And we know that hydrochloric acid that we dissolved must dissociate into a proton plus Cl minus. Okay, and so therefore we can write that the concentration of protons must equal the concentration of chloride anions plus the concentration of hydroxide anions. I can simplify this in two ways. I can say here that the chloride concentration must equal about 0.001 molar, because after all, the hydrochloric acid is completely dissolved, right? And therefore, I can write that the proton concentration must equal 0.001 molar plus the concentration of OH minus. And if I wanted to, I could use this equation for OH minus so that what I would end up with, I could replace the concentration of OH minus with 1 times 10 to the minus 14 molar squared over the concentration of protons. And if I do that and I plug that in, then I'm going to have a quadratic equation in protons, right? And I could solve the quadratic. Okay. Or I could just make some reasonable assumptions. I can assume, for example, that since it's an acidic solution, right, if it's an acidic solution, then clearly you would agree that the concentration of OH minus is a lot less than 0.001 molar, right? So the concentration of hydroxide anions is a lot less than the concentration of chloride anions. So I can just neglect this, right? Okay. So I can therefore say, so the proton concentration, therefore, is going to be approximately equal in this case to the concentration of chloride anions, which is equal to 0.001 molar, right? And therefore, the pH is now 3. So the pH started out for H2O, started out at 7, and then after I added 1 millimolar of hydrochloric acid, it dropped to 3, and I had a change in pH of 4 units. Okay? Easy. Super easy if I assume complete dissociation. Super easy if I assume complete dissociation. The problem is that for many weak acids, I can't make that same assumption. Okay? So what happens if I have a weak acid? Okay? For a weak acid, I have to say that HA now is largely undissociated. It will dissociate into H plus A minus. Okay, and I'm going to now define something a little different. I'm going to say my equilibrium constant here is, of course, the proton, the conjugate base, divided by HA. And I'm going to give this a special name. I'm going to call this Ka for K acid. And here I have to apologize for the field of chemistry as a whole. Okay? So I forever was confused about this because I always learned that Ka meant K association and Kd meant K dissociation. You guys ever seen that? And then people start talking about Kas, and I couldn't figure it out because it looked to me like this was a dissociation constant. So I apologize for chemistry. Okay? When you see Ka, you have to ask the person that's explaining it, do you mean K acid or K association? And in this class, I promise you, whenever I use Ka, I'm going to be talking about K acid. Okay? So let's do an experiment now with acetic acid. And what I want to do with acetic acid is something like we did with hydrochloric acid, but a little different. What I want to do here is we're going to take acetic acid and we're going to, which is, as I mentioned to you, right, it's basically, I guess I should write it out. Acetic acid is this. It dissociates to some extent into acetate, CH3, CO- plus a proton. And its Ka is 1.75 times 10 to the minus 5 molar. Okay? So it's a small number. It only dissociates to a small amount. So the experiment that I now want to do is I want to add 10 millimoles of acetic acid, which I'm going to abbreviate like this, HAC. That's acetic acid. Into 1 liter final volume of H2O. And I want to ask, what's the final pH? Now to do this, I need 4 equations. Okay? Because I can't assume complete dissociation. Okay? So 2 of the equations are determined by equilibria. Okay? So 2 correspond to equilibria. And those equations are that the concentration of protons times the concentration of acetate anion is simply equal to 1.75 times 10 to the minus 5 molar times the concentration of acetic acid. We'll call this equation 1. And of course, the second reaction is nothing other than the ion product of water. Right? That's just the concentration of protons times the concentration of hydroxide anions has to equal 10 to the minus 14 molar squared. That'll be equation 2. Okay? Now I have to add a proton balance. So here's my third equation, proton balance. Clearly, you would all agree that the total concentration of protons here has to equal the two sources. It has to equal the acetate anions plus the hydroxide anions. We'll call this equation 3. And then the last equation is a mass balance on the acetate. Okay? You know that I added a total of 0.01 moles into a liter, and this has to equal, therefore, the concentration of acetate anion that I finally end up with, right, plus the concentration of acetic acid that hasn't dissociated. And we'll call this equation 4. Now, I have 4 equations and I have 4 unknowns. Protons, hydroxide anions, acetate anions, and undissociated acetic acid. 4 equations, 4 unknowns. I could plug and chug. Or I could make some assumptions. So, 4 equations, 4 unknowns, plug and chug. Or let's make some assumptions. So the first assumption I think we can make is that it's an acidic solution. And that means that we can probably say that the concentration of acetate anion is greater than the concentration of OH minus of our hydroxide anions, right? And that means, therefore, we can write that the proton concentration total, which we said here was in equation 3, was equal to acetate plus OH minus, is probably about equal to just the concentration of acetate anions. Okay, that doesn't seem so unreasonable. Okay? The second assumption we can make is I told you that acetic acid is a weak acid. Okay? So it doesn't dissociate very extensively. So maybe that means that the concentration of acetic acid at the end isn't that dissimilar from what I added because not a lot of it dissociated. So I can say that maybe this is about equal to the amount I added, 0.01 molar. Okay? And so with those assumptions now, I can go back to equation 1. This is the equation that I'm going to use. And let's see what I get. I can say from that the concentration of protons times the concentration of acetate anions equals 1.75 times 10 to the minus 5 molar times the concentration of acetic acid. I already told you that I'm going to make the assumption that this is 0.01 molar. I'm going to pretend it didn't dissociate. And because the proton concentration is roughly equal to the acetate concentration, I can replace this with proton concentration. This is about also equal to the proton concentration. And so now I end up with a proton concentration squared that's equal to 1.75 times 10 to the minus 7 molar. Okay? And a concentration of protons, if I take the square root, that's going to equal 4.18 times 10 to the minus 4 molar. And if you take the minus log 10 of the pH here will now be 3.38. Okay? So we took H2O, which had a pH of 7.0, and we dropped it here with a change in pH of 3.62 pH units. Now stop for just a moment and think about what we did. We added 10 times as much acetic acid as we added hydrochloric acid. Remember here with hydrochloric acid, we added 1 millimole, and that dropped us from 7 to 3. With acetic acid, we added 10 millimoles, 10 times as much. But that didn't drop us to 3. It didn't quite make 3. It wasn't as good an acid. It did not dissociate to the same extent. Okay? Now, let's test our assumption. Was it fair to make these assumptions that we did? Let's see. Let's test our assumptions. We said at the beginning that the concentration of acetic acid in the end was going to be about equal to what we started with, which was about 0.01 molar. Okay? Now we know from this that the concentration of protons is equal to the concentration of acetate anions, which is equal to 4.18 times 10 to the minus 4 molar. Okay? And so if I now calculate what the real concentration of the undissociated acetic acid is, it would be 0.01 molar minus 0.0004 molar about, I guess I should 0.18, yep, 4 molar, which is equal to 0.0096 molar. So our assumption was accurate to within 4%. There are two things that we have to get to from this thing that we've worked out here. The first is I want to talk a little bit about Ka, this acid, this K acid. And as you know, it's a fundamental chemical property of the acid, right? It's determined, the dissociation extent is determined by the chemical properties of what makes the acid. The second thing that I want to tell you about is that if we know Ka and pH, this is the important thing. If we know Ka and the pH, then we can calculate the relative ratios of the undissociated acid, HA, and the conjugate base A minus. Okay? Let's see how to get there. Okay? Now, you would agree that we agreed that Ka, being a dissociation constant, reflects H plus A minus over HA. This is just a statement of chemical equilibria. And I can take the minus logarithm to the base 10 of this side and of this side, and look at what I get. That the proton concentration is equal to Ka times HA divided by A minus. And now I'm going to take the minus log 10 of this. And that will give me minus log 10 of proton concentration is the pH. And that is therefore equal to minus the log 10 of the Ka, which you guys told me was the pKa, right? And then it's going to be minus the log to the base 10, right, of HA over A minus. And I'm going to rewrite this like this, that the pH is equal to the pKa plus the logarithm of A minus over HA. All I've done is to remind you that if I flip the numerator and denominator, that's the same as changing the sign of the log. Okay? And this equation you will use again and again and again in your career. This equation has a name, right? It's called, and again, this equation is called the, anybody know? Yeah, of course. This is the Henderson-Hasselbach equation. And the way I want you to think about it is, says that the pH is the pKa plus the logarithm here of the proton acceptor concentration divided by the proton donor concentration. Okay. Okay. Well, what does it tell us? It only tells us the ratio of that conjugate acid of the acid to conjugate base. It doesn't tell us the absolute amounts. It only tells us about the ratio. So let's go back now to acetic acid. You know that acetic acid, I've told you, has a Ka of 1.75 times 10 to the minus 5 molar. And if you take the minus log to the base 10, this means that the pKa is 4.76. That's the pKa for acetic acid. Okay? And what this means is, what does this tell us about acetic acid, a solution of acetic acid at different pHs? Okay? So suppose I have a solution of acetic acid, and the pH of that solution is 4.76. Okay? What does that mean? Well, 4.76 is the pH, and that equals the pKa, 4.76, plus the logarithm, right, of the acetate anion divided by the amount of undissociated acid. So what's the ratio of the conjugate base to acid here? 1, right? This ratio must equal 1 because the log to the base 10 of 1 is 0, and 4.76 plus 0 is 4.76. So what this means is my solution of acetic acid at pH 4.76 has an equal amount of the undissociated acetic acid and the acetate anion. If the total amount of acetic acid in the solution is 1 molar, then I have half a molar of acetic acid and half a molar of the acetate anion. If the solution is 10 millimolar, then I have 5 millimolar of the undissociated acid and 5 millimolar of the acetate anion. Okay? What about, suppose I'm 1 pH unit greater than the pKa. Okay? So now I make my solution, and it has a pH of 5.76. Okay? Well, what does that mean? Well, that has to equal the pKa plus the logarithm of the acetate anion over the undissociated acid, HAC. Okay? So what does this ratio now have to be? Give somebody. What's the ratio? 10 to 1, right? This ratio has to be 10 to 1 because the log to the base 10 of 10 is 1, and 4.76 plus 1 last time I checked was 5.76. And so this means that when I'm 1 pH unit, this is what I want you guys to write down and remember. When I'm 1 pH unit greater than the pKa, 90% of what I have is the anion. Okay? This means at 1 pH unit, I'll write it down. 1 pH unit greater than the pKa, I'm 90% unprotonated. Okay? Suppose I'm 2 pH units greater than the pKa. Okay? Then I'm talking about 6.76 equals 4.76 plus the logarithm of the acetate anion divided by the undissociated acetic acid. And this ratio, therefore, would be what? 100, right? The ratio is 100 to 1. And this means that when I'm 2 pH units above the pKa, now I'm 99% unprotonated. Let's do one more example. Consider 1 pH unit below the pKa. Okay? So that means my pH is 3.76. That must equal 4.76, the pKa of acetic acid, plus the logarithm of the acetate anion concentration divided by the undissociated acetic acid. And once again, you're going to tell me, choose a different color, what the ratio of this must be. So what's the ratio equal to? I can't hear you. 1 to 10, right? Because last time I checked, the logarithm of 1/10 is -1, And 4.76 minus 1 is 3.76. And so the punchline that I want you to remember here is that at 1 pH unit below the pKa, 90% is protonated. 90% of the acid is protonated. By the same token, if I'm 2 pH units below the pKa, what percentage of the acid is going to be in the protonated form? 99%.